Okay, we're going to start our first real lecture on molecular biology. And nucleic acids are really the components that all molecular based tests are based off of. So, nucleic acid biochemistry is really an important concept to understand. Now, this should all pretty much be a review of things that you covered in a general biology course. However, some of you may have taken a biology course a long time ago, so this is a nice overview. If some of this material is not familiar to you, you may want to go to a molecular biology or cellular biology textbook and review the nucleic acid chapters in the textbook. If you've purchased the recommended textbook for this course that's optional, you can read the RNA and DNA chapters, chapter 1 and 2, which covers all of the material that we will be discussing in this lecture. The objectives for the nucleic acid biochemistry lecture include the identification of the cellular elements involved in growth. At the end of this lecture, you should be able to list the functions and components of the nucleus. There are several terms that you should be able to define, such as nucleic acid, nucleotide, nucleoside, DNA, RNA, chromosome, chromatin, and gene. You should be able to list the nucleotides that make up both DNA and RNA, compare and contrast DNA and RNA, and outline the steps involved in DNA replication. We're going to begin talking about the bigger picture, or the cell. And as you all know, there are many different types of cells, and cells are very specialized. Every cell has a specific function depending on its location in the body. So there are many components that make up a human cell. And we're going to focus a little bit more on the nucleus because that's where the DNA is located. So the elements of a cell that are involved in growth, that would be the nucleus, the ribosomes, the endoplasmic reticulum or ER, Golgi, vacuoles, and centrioles. The elements that are involved in movement of the cell are the centriole, cytoskeleton, actin and tubulin proteins, and flagella or pseudopodia, which are external elements outside the cell. Cellular elements that are involved in metabolism or generating energy. These are the membranes, vacuoles, mitochondrion, chloroplasts, which are found in plant cells, and the cytoplasm. So the nucleus. The nucleus stores the genetic information that the cells need. So the nucleus is really important for the cell. Subcomponents of the nucleus are the chromatin, chromosomes, nucleolus, and nucleoplasm. Our nucleic acids, which is what we're going to focus on in this lecture. Nucleic acids are large molecules and they have two main components. That's a sugar and a phosphate. There's a sugar and phosphate backbone, and that backbone is attached to a nucleotide base. There are 4 nucleotide bases that make up nucleic acids. Each nucleic acid contains millions of nucleotides that are bonded to it, and the order of these nucleotide bases is what the information that the cell needs to carry out specific functions, and that's going to become more clear in the next 2 lectures. So the nucleic acid backbone, we have a sugar, phosphate, sugar, phosphate, sugar, phosphate over and over and over again. Attached to the sugar is a nucleotide base, and that nucleotide base could be one of 4 possibilities. And depending on the order of those nucleotide bases, it's going to give you a different function. So DNA. DNA is what we're going to discuss first in this lecture. DNA is deoxyribonucleic acid, and it's the genetic information storage system. So all of our cells need DNA in order to function. So a little bit of history of DNA. Mendel, considered the father of genetics, is the person that described the unit character of genetic traits, and this was done in 1865. In 1868, Miescher isolated what was called at that time nuclein. In the 1920s, Levine identified the chemical components of DNA. Some definitions that are very important. A deoxynucleoside is a combination of a deoxyribose and a base. And a deoxynucleotide is a combination of a phosphate, a deoxyribose, and a base. So very similar, slightly different there. Deoxynucleotides are the building blocks of nucleic acids, and they're used to store and transfer energy in the form of ATP. So DNA is a polymer of nucleotides. Nucleotides are phosphorylated nucleosides. Nucleosides, we already said, are comprised of a ribose sugar and a nitrogen base. There are 4 common nitrogen bases. 2 have a purine ring structure, and these are adenine and guanine. And 2 of the bases have pyrimidine ring structures, and these are thymine and cytosine. So the order, or what we call the sequence of the nucleotide bases in a DNA polymer, is the coding information. And we'll discuss more about that when we talk about translation. So this is just a schematic of your nitrogen base, your deoxyribose, which make up your nucleoside, and your nucleotide. So deoxyribonucleotides. You have deoxyguanosine phosphate, which is your guanine, DG, your deoxyadenosine phosphate, your DA, deoxythymidine phosphate, and deoxycytidine phosphate. All of these come together in DNA in a different sequence depending on what area of the genome you're looking at and what individual you might be looking at. So that nucleic acid sequence is what we're going to really look at to determine if someone has a disease or not. So one mutation or variation in that nucleic acid sequence could be the difference between having a normal sequence and a sequence that's going to lead to a disorder. So more history. In the 1920s, Griffith discovered what's called the transforming principle. In 1944, Avery, McLeod, and McCarty showed that the transforming principle is actually DNA. In 1952, Hershey and Case showed that DNA carries the genetic information. And Chargaff, we're going to talk about Chargaff a little bit more in detail, found that the number of purines is not equal to the number of pyrimidines. So what Chargaff showed is that A plus G divided by T plus C equals 1. So the ratio of A's and G's and T's and C's equals 1, and that was done in the 1950s. So DNA exists as 2 polymers joined together by hydrogen bonds. So your nucleotide bases are joined together by hydrogen bonds, and A always binds with T, and C always binds with G. There are 2 hydrogen bonds between A and T, and there are 3 hydrogen bonds between C and G. Your 2 polymers, those 2 backbones, form a double helix. So that's why your C bonds with G, your A bonds with T, and your double helix, you have one backbone in the 5 to 3 prime direction, and the other one is in the opposite direction, 3 prime to 5 prime direction. So they are anti-parallel to one another. So that parallel and anti-parallel, that's because DNA has polarity, and this is based on that backbone, that phosphodiester backbone structure. So when you're reading a DNA sequence, you always read it in the 5 prime to 3 prime direction because that's the polarity that DNA has. So DNA, we already said, is the genetic information. You find it in the nucleus of all living cells. Deoxyribose is the sugar in that backbone. There are the 4 bases, the nucleotides, that make up the DNA. These are adenine, guanine, cytosine, and thymine. Adenine and guanine are the purines. Cytosine and thymine are the pyrimidines, and you should know which ones are purines and which ones are pyrimidines. So once again, purines, adenine and guanine, pyrimidines, cytosine, and thymine. So your DNA, you have your backbone, sugar, phosphate, sugar, phosphate, sugar, phosphate, continuously. Attached to the sugar is your nucleotide base, and that could be either your A, T, G, or C. So the composition of whether it's an A, T, G, or C is going to vary depending on what gene you might be looking at. However, keep in mind the amount of adenine equals the amount of thymine, and the amount of guanine equals the amount of cytosine, because those are what bind together in your double helix. So DNA is double-stranded. Your nucleotide bases form hydrogen bond pairs. So we already said adenine binds to thymine, guanine binds to cytosine, and these are done through hydrogen bonding. So you have your phosphate, your sugar, your phosphate, your sugar, your phosphate, your sugar. Your nucleotide attaches to the sugar. So we mentioned Chargaff's rule, or Chargaff in our history of DNA. And Chargaff's rule is that A equals T and G equals C. We'll go through that in a little bit. So what Chargaff found is that there's a regularity in the ratio of nucleotide bases. So he studied DNA from various species and found that the number of adenines equaled pretty much, not exactly, but approximately to the number of thymines, and the number of guanines equaled the number of cytosine. So, as an example, in human DNA, you might find that in a certain gene, your A is in the percentage of 30.9, your T is 29.4, your G 19.9, and C 19.8. So they're not absolutely exact, but they're very similar. So A equals T, G equals C, and this is how Watson and Crick discovered the structure of DNA, realizing that the proportion of A's and T's and G's and C's, that ratio, was very important. So that's how they figured out that A always binds to T, G always binds to C in that double-stranded structure. So again, your percentage of A plus G is equal to the percentage of T plus C. Here's your double-stranded DNA. Again, sugar, phosphate, sugar, phosphate. Your nucleotides always are going to come off of your sugar backbone. Those nucleotides can be either T, A, G, or C. And on the opposite strand If you have a T on one strand, you are going to have an A on the other strand, right on the opposite across from it. And the nucleotide bases are bonded together through hydrogen bonds. If you have a C, you're always going to find a G on the opposite strand. If you have a G, you're always going to find a C. So you should know, if you were given one strand of a DNA sequence, you should be able to figure out what the opposite strand sequence should be, because they're complementary to one another. So once again, your double-stranded DNA, you have your sugar, phosphate, your nucleotides coming off of the sugar. Your C is going to form a triple hydrogen bond with G, and your T is going to form a double hydrogen bond with A. Once again, your triple hydrogen bond, your double hydrogen bond. And this is just more of a chemical structure of one strand of your DNA backbone. So the building blocks, those nucleotides, are the building blocks of your DNA. They're absolutely essential. So you have your DATP, or deoxyadenine triphosphate, DGTP, deoxyguanine triphosphate, DCTP, deoxycytidine triphosphate, and DTTP, deoxythymine triphosphate. So in 1953, James Watson, Francis Crick, Maurice Wilkins, and Rosalind Franklin all determined the exact structure of DNA. Watson and Crick received the most credit for that, but it was a group effort. They found that DNA forms that double helix, and that double helix wraps around one another, so it's in a form of a coil. Also, DNA must be able to make exact copies of itself. So our cells are constantly undergoing division. We're constantly making new cells. So in order to make new cells, we have to replicate that DNA in the nucleus of our cell. So our DNA is able to replicate. And the next lecture that we do is going to be DNA replication. Here's a schematic of your DNA double helix. So not only do you have two backbones that are in an anti-parallel direction with your nucleotide bases hydrogen bonded to one another, but it's all in a coiled form. So each strand, so the backbones, are linked together through phosphodiester bridges. So that's the backbone, so your double helix backbone. Your nucleotides are linked together through hydrogen bonds. The 3' carbon of one ribose in the deoxyribonucleotide binds with the 5' carbon in the ribose of the opposite deoxyribonucleotide. That's your anti-parallel structure. And your polynucleotide, or your DNA, is always red from the 5' to the 3' end. So you don't need to know, the distance between twists and things like that, but just keep in mind that, we simplify the DNA structure, but it's actually very complex. And if you were to link up all the molecules involved in a DNA double helix, this would be what you would come up with. So DNA replication, we are just going to outline the steps of DNA replication in this lecture, because our next lecture is going to be focused completely on replication. So we'll go into a little more detail in the next lecture. So DNA replication, you have your hydrogen bonds between your nucleotide bases. These must break so that your double helix breaks apart. Those two strands separate. Then you get new bases that are complementary to each strand, and they are paired back up, so now you have two identical double-strand DNA molecules. So let's look at a picture of this. You have your coiled DNA double helix. Your nucleotide bases have to break apart, and then you have to generate new bases based on the sequence of the bases that are there. So you're going to get complementary bases, hydrogen bonding to one another, and your second backbone. So you're going to have then from one double-stranded DNA, you're going to get two double-stranded pieces of DNA. So here we have our old strand. It's going to break apart. You get your new bases that are going to attach in a complementary fashion, and then you get two new strands that are identical to the previous old strand. So there's a video. If you copy this website and paste it into your Explorer or Firefox or whatever you're using, it'll take you into a video of DNA replication so you can really see this. I find videos very helpful. I'm a very visual person, so if I do put in a video, sometimes I will post the actual video, but other times I'm just going to give you the link and you can go in and look at that video. Okay, so that's DNA. Now we're going to talk about RNA. RNA is very similar to DNA. There's just a couple of very small but very important differences, and you should know the differences between DNA and RNA. So DNA and RNA. DNA, we said, is double-stranded, that nice double-stranded coiled helix. RNA is single-stranded. DNA is deoxyribose sugar. RNA is ribose sugars. DNA contains ATGC. RNA contains A, G, C, and U. So instead of thymine that is in DNA, RNA has uracil. So wherever there would be a T, if you were looking at RNA, there'd be a U. There are different types of RNA. There's messenger RNA, ribosomal RNA, transfer RNA, heteronuclear RNA, small nuclear RNA, microRNA, short interfering RNA, double-stranded RNA. Double-stranded RNAs are usually found in viruses. So in human cells, RNA is a single-stranded molecule, but there are viruses that are double-stranded RNA viruses. So the most important types of RNA in a human cell are your mRNA, your rRNA, and your tRNA. And those are in red. So replication. RNA has to replicate just like DNA replicates. Remember, our cells are constantly dividing, so you have to replicate the nucleic acids within the cell. So with RNA replication, there's no priming that's involved. And again, when we talk about replication in the next lecture, this will make more sense. RNA replication has more initiation sites than DNA replication. RNA replication is slower. It has lower fidelity and it's more precessive. So those are the slight variations between RNA and DNA replication that we will discuss in more detail in the next lecture. Okay, RNA replication has an initiation step that begins at a promoter. So there's a sequence in the RNA that is what's going to say, hey, let's start replicating right here. You need an enzyme during replication. So for DNA, you have DNA polymerase, and RNA, you have RNA polymerase. So once again, we will talk about replication in much more detail in the next lecture. Histones. So histones are important with nucleic acids. Histones allow your RNA polymerase enzyme to have accessibility to that DNA so that you can get transcription and translation to occur. So those are going to be our next few lectures: replication, transcription, translation. So what histones are these little structures that DNA can wrap around, and it allows DNA to be more tightly packed inside the cell. So the histone code, you could have different modifications of histones. So you can have acetylation, phosphorylation, methylation, ubiquitination, ribosylation, and glycosylation. And these are all important modifications depending on what process is happening in the cell. So the RNA structure. Just like DNA structure, you have a backbone. So you have that sugar, phosphate, sugar, phosphate. The sugar is ribose in RNA. It's deoxyribose in DNA. Once again, you have your 4 nucleotide bases: adenine, cytosine, guanine, and in RNA you have uracil. In DNA it would be thymine. RNA is single-stranded, whereas DNA is double-stranded. So once again, your RNA, sugar, phosphate, sugar, phosphate, sugar, phosphate backbone, and you have your nucleotides coming off the sugar. 4 nucleotides, the only difference is the uracil instead of the thymine. The function of RNA. So DNA is that genetic information. What does RNA do? RNA is what relays that information that's inside of cells. So RNA is able to come out of the nucleus and go to other parts of the cell. So RNA is absolutely essential for the production of proteins. Where can we find RNA? Well, it's synthesized inside the nucleus where the DNA is. So you need your DNA in order to synthesize your RNA, and the DNA is found in the nucleus. Well, then your RNA moves out into the cytoplasm, and that's where proteins are going to be made. So you have your messenger RNA, which carries that genetic information based on the DNA out of the nucleus. You have your transfer RNA, which decodes that genetic information, and you have ribosomal RNA that constitutes your ribosomes, which are very important in your protein development. You also have catalytic RNAs that are involved in different reactions inside the cytoplasm of the cell. So transfer RNA. The only thing about transfer RNA is that it's the smallest RNA. It's very tiny. It's usually made up of only 70 to 90 nucleotide bases. And what does transfer RNA do? It transports amino acids to the site for protein synthesis. And this is going to be made very clear when we talk about transcription and translation. So RNA. We already said RNA is single-stranded, so it's a single polynucleotide as opposed to DNA. However, RNA is not a linear structure. We already said viruses can have double-stranded RNA, just like human cells have double-stranded DNA. But in human cells, RNA is single-stranded, but it isn't linear. What RNA does is it folds up on itself, forming these different structures that look like they're double-stranded. However, RNA is single-stranded. So they form these double-stranded structures based on complementarity. So just like in DNA, your A is always going to bind with T and your G is always going to bind with C. In RNA, it's single-stranded, but you have regions that are going to fold up on itself based on complementary nucleotides. So here's an example of some of the secondary structures you can get in RNA. You can get what's called a stem, where you're going to have A's and U's binding together and C's and G's binding together. You can have hairpin loops, pseudoknots, bulges, internal loops, branch loops. So you can have these little double-stranded looking secondary structures, but it is not double-stranded. It's single-stranded. It folds up on itself. RNA synthesis. So you use a portion of your DNA to make RNA, and there are nucleotide sequences that are going to tell where you should start making RNA and where RNA should stop being made. So these would be called promoter sequences and termination sequences. So a gene, usually you're going to generate or make RNA off of DNA that is in a gene. So a gene is a segment of DNA that codes for a protein, and proteins are what is the actual function. So you have a gene for every single solitary thing that a cell needs to do. So you have a gene that's going to give a trait such as skin tone, eye color, hair color, etc. Your chromosomes are ordered structures of single double-stranded DNA molecule that's wound up very, very, very tightly. So your chromosome allows all that DNA, millions and millions and millions of bases, to all be compacted very, very tightly and held inside the nucleus. So you can actually calculate the length of DNA. You do not have to do this. You will not be asked to do this on any quiz or exam. I just wanted you to know that there is a mathematical calculation that you can do to determine the length of DNA. So keep in mind that if you were to take your genomic DNA inside your nucleus and stretch it all out, it would be the length of 70 trips back and forth between the Earth and the Sun. So DNA is wound very, very, very tightly into chromosomes so that it can fit inside the nucleus. So your chromosomes are your DNA strands that are coiled around histones. We mentioned histones a little while ago. Your human cells are diploid, which means there's 2 full complements of the human genome in the cell. Each copy, since there's 2, is about 3.2 billion nucleotides, and it's organized into 23 chromosome pairs. You have 1 maternal and 1 paternal, which makes a pair. So in human cells, there's 23 chromosome pairs. In cats, there are 30. In male honeybees, there are 8. So depending on what cells you're looking at, you have different numbers of chromosomes. Chromosomal arrangement. So you have your huge genome that's in the teeny tiny space of the nucleus of a cell. So that's why chromosomes are important. That arrangement of chromosomes, that 1 maternal and 1 paternal chromosome that make up your pair, that's how you get Mendelian genetics. And we are going to talk about genetics in a couple of lectures. Different genes are located on different chromosomes, and chromosomes can recombine from one generation to the next, and this is where you might get a mutation that can form. Chromatin is your nuclear DNA and all of the proteins, such as that histone where your DNA wraps around it. So all that makes up your chromatin. And your cells during cell cycle development, your chromatin is going to change in its condensation. You have euchromatin. These are genomic regions that have a lot of genes, and genes are what code for proteins, and these are not as compactly organized. You also have heterochromatin. These are regions of your genomic DNA that don't have a lot of genes. They're gene poor, and they're very densely packed regions. So coiling. We already said DNA coils around one another, wraps around histones, and like I already said, you don't need to know the different lengths of each coil and things like that, but your chromatin has a bead on a string structure. So your DNA wraps around a histone and then wraps a little more around another histone, so it kind of looks like a string of pearls. So this is a cute cartoon to wrap up our nucleic acid lecture. And really, your nucleic acids, especially your DNA, your nice double-stranded DNA, is kind of a long, twisty, rope laddery looking thingy. And that's a perfect description of DNA. So our next lecture, we are going to talk about replication, then we're going to talk about transcription and translation, and all of these concepts that we talked about in this lecture are really going to come together in the next few lectures.